question is, could you use the diagnostic pathology image that is used to make the diagnosis in the first place? And is there information content in that digitized H&E to actually predict this without having to send the material out for testing? And so we assembled a cohort of 5,000 patients that had digitized H&E pathology slides, as well as the Oncotype DX score and the synoptic pathology report. And we built a vision transformer that basically takes the pathology image and decomposes it into small tiles, and then creates a vision transformer from that. And then on the bottom, we took a text-based tokenizer and built a language transformer, integrated those two things. So the modes here are the pathology image and the actual synoptic report. So you have a text and image coming together. And so the results are that we can actually do fairly well in predicting the Oncotype DX score. And so what is shown on the left is actually the green is the language transformer alone, so that's the text alone. The brown is the vision transformer, and the purple is the combined. So you can see that the combined, the multimodal predictor, does better than either one alone. And overall, actually, the AUC score is quite good. It's actually 0.89. And so we can actually, just with diagnostic data that's actually procured in the provision of care, start to predict what this very expensive test actually predicts. And so for triaging, for example, we can, at the tails of this distribution, I think we can actually think about a path to replacement of that and make decisions much earlier than we otherwise would be able to do. Okay, great. So we've done this in a few other different contexts, and just listing these papers here for interest. And so these are all, I think the breast cancer study is the biggest, 5,000 patients, not small. But all of these are sort of limited by scale to some extent. So we're in the hundreds of patients on the two papers on the left. We're getting into the thousands of patients in the two papers on the right. But we started to think about, well, let's try to do this at scale with all the data resources at MSK. And so in parallel to the genomics initiative at MSK has been running for 10 years, there's been a scanning effort to digitize the archive of pathology specimens, and that's been taking place. And so we decided that we should try to marry up all the patients that have received MSK impact sequencing, and this is a panel-based sequencing test that comprises about 500 genes, is tumor normal, and most solid tumors that come in the door will have this testing. And so we have now a database about 100,000 patients with this sequencing test done. We have all the raw data, we have the mutation data. And so then we went into this digital archive that's been growing from the pathology department and layered on this information on top of these 80,000 patients that have genomic testing. So now the modes are digitized histopathology and genomic testing. And so we started to think about, can we build larger scale models that can start to learn the principles of how do alterations in the genome actually impact what you can read out in a digitized H&E. And so this is a collaboration of the Warren Alpert Center for Computational Pathology. And so we can start to embed these images using pre-trained transformer models. And the first thing we wanted to try to do is see if we could recapitulate diagnosis. And so typically the literature maybe has very coarse representations. The previous attempts on this have very coarse representations of tumor types, maybe 18 tumor types. Of course, we know that the granularity of histopathology will get us into hundreds. In this case, we encode that with something called OncoTree. It's just an ontology of histological subtypes. And OncoTree contains 163 granularly defined histological subtypes. Okay, and that's I would say a community standard. It's used at MSK, but it's also used in AACR Genie and some other places. And so then from that, from those granular histological subtypes, then we can start asking questions about what is the prevalence of particular types of mutations that are present in those granular histological subtypes to try to get a better understanding of maybe what's driving those subtypes to begin with. Okay, so that's the basic setup. And so you can actually look at the oncotree subtypes with a graph. And here we sort of have this notion that the histological subtypes have some distance associated with them, and that can be encoded by their developmental lineage is the most informative thing. And so we can represent this graph and start to group these subtypes together, but basically by their tissue lineage. And you can see that the neuroendocrine space is in yellow there is grouped together by distance. The melanomas are grouped, the sarcomas are grouped, et cetera, nervous system, et cetera. So that makes sense. And this is just, this isn't doing anything new. It's just encoding that information into a graph structure. So then what we can do is we take the 80,000 histological images, we embed them using this pre-trained transformer model. And in that embedding space, we can ask, just without any information, can the embedding space capture something that looks like what's on the left? And so then now here, each data point is one of these images from a tumor. And they group quite nicely according to the major lineages. And so the color coding here is basically the coarse grain histological types, but at a higher level. And you can see that, for example, the GI tumors mostly track together, the CNS tumors track together, the soft tissues are together. So there's information, content in these images that represent their histological lineage. And that itself may not be so surprising, but it's really quite gratifying that in a very automated way, without knowing anything about the histological labels, we can recapitulate this with a pre-trained transformer model. Okay, and so the scale of this is 163 cancer types, about 80,000 tumor samples, 70,000 patients, and ultimately what goes into this model is in the range of 5 billion tiles that we decompose the images into these little tiles. So that's the scale of this data, and actually to fit this, because we're using a pre-trained transformer model, we don't have to do the end-to-end training. We're just pulling that model down off the internet and actually computing the embedding space. This isn't actually as big a compute lift as it sounds like. So if you were to do end-to-end training, this would take a long time. But we can do this in a matter of a weekend, actually, using internal GPUs. All right. So where we think this is potentially helpful is improved refined diagnosis, making associations with genomic alterations, and potentially being able to infer clinically actionable biomarkers. So just from a histology side, you could potentially stratify patients onto a given therapy based on what the information content in that image. And that's without having to do the genomic testing in the first place. So you can imagine that would be quite useful in resource-constrained environments or where there's time pressures and time constraints to make a decision. So this figure here shows that a lot of the annotations in the clinical record are actually of this coarse-grained nature, where these granular histological subtypes are not specified in the pathology report. And so you can imagine that here you have breast cancer, the BRCA on the top, and then you have breast cancer not otherwise specified. And so that's what those three rows at the top are. And so when we actually try to classify them into their granular subtypes now on the x-axis, you can see that they actually really nicely can be broken down into ductal and lobular carcinoma, et cetera. And so you start to get the histology right from the image where the pathologists themselves had not entered that information into the record. So then you get a little bit more precise histological information. And you can see that again in the colorectal, rectal cancer, the blue block there, we get this very nice decomposition into the histological subtypes, et cetera, et cetera. And you can see that there's really kind of not that many off-target entries in this matrix. There are some, but by and large, the histological subtypes that this model predicts are within the coarse grain subtype to begin with. So that's actually really quite gratifying. It's not classifying something that's wildly different. All right, and then so now we can get into a little bit more detailed. So this is a set of tumors that are the stars here represent sarcoma not otherwise specified, and you can see that these stars actually almost fill the whole space of the granular histological subtypes that are annotated here. And so we can start to now take this diagnosis that was originally sarcoma not otherwise specified and start to put them into spaces that are much more precise. And so that's helpful. And we now have a sarcoma pathologist that's helping us to actually validate this. But we've seen in anecdotal cases that, yes, on review, it actually should have been called the more precise granular subtype that the model is actually predicting. So for diagnosis, I think there really is a potential here for refinement in an automated system. If any of you had to go, I won't be offended. Okay. Hope someone's okay. So now we can go into some other ideas now, bringing into the mutation data. So now what we have is a representation of this embedding space in 2 dimensions, and again each point is a tumor, and so here are subtypes of renal cell carcinoma, and in one subtype we expect that they should have von Hippel-Lindau mutations, and that's just well known in the literature. And so when we overlay these mutations onto spaces that are occupied by what the model classifies as much more granular histological subtypes, we can see indeed that all these blue points, these VHL altered, they all are overlaying on this group of tumors that are classified as clear cell renal cell carcinoma, and the other subtypes don't have those mutations. So that tells us something that not only is a histological subtype predictable, but then now you can start to associate that the expected mutation rate is actually essentially validates this prediction. So this is where now overlaying the genomics information is really quite powerful. So then we took this one step further and started asking, okay, well, can we train a model now that can predict activating or actionable oncogenic mutations? Now we're in the lung cancer regime and keep 1 mutations. And so what we can see here is that there is if we now look at the specific mutations across the x-axis, you can see that some of those mutations have basically track with what we know are the oncogenic mutations in that disease. These other mutations are what we call variants of unknown significance. They may not have been studied. They may not have functional data that goes with them. But we can make a prediction that these mutations that are towards the left of this plot are likely to have some sort of functional significance, and the ones on the right are probably not going to have functional significance. So we can start to interpret mutations by integrating them with the embedding space of the pathology information. So that helps us maybe conduct some hypotheses that could then be tested in the laboratory. So overall, I think this whole engine allows us to ask many limitless questions, really, about the integration between genomics and histopathology. And I think going forward, what we want to do is now think about an embedding space that encapsulates both. So the joint embeddings, where we learn these models where you can reduce both the genomics and the histology into a combined joint embedding space. And we think that this is going to have some new representation for diagnostics and will help with risk stratification and outcome predictions. So at this point, I'll just summarize where we are. So I've shown you some examples of where integrating multimodal data improves risk stratification. The pathology embedding spaces certainly can refine cancer diagnostics. And one part of this study that I didn't show you is about 5% of cases are diagnosed as cancers of unknown primary. And we can actually start to infer what's the likely primary tumor from these spaces as well. And then lastly, these pathology embedding spaces do correlate with functional mutations. And I did put up the preprint link in a few slides ago. Happy to share it again. But all of the details of the study are written up in that space. Okay, so now turning to the right-hand side of that overview slide about discovering new tumor biology. So I mentioned that the technology advances are really kind of formidable and ferocious, where we now have ability to, with spatial transcriptomics information, is to in situ, for example, measure the activity of tens of thousands of genes within one cell while the tissue is intact. So that's driving, I think, incredible pace of discovery. And what I'll show you is just some of our work in those spaces, both in single cell genomics, tumor phylogenetics, the cellular phenotype work, and what we can, the types of things that we can elucidate from using these measurements in a multimodal fashion. So when I went to Memorial now almost 8 years ago, we set up a prospective collection in ovarian cancer. And so part of my lab studies high-grade serous ovarian cancer and the evolution of that disease. And so we set up this collection protocol with the surgeons where at primary debulking surgery or at interval debulking surgeries, we collected material from across the peritoneal cavity, and you can see that's depicted there in the diagram, and then prepare these tissues for a single cell analysis. And this was by generating single cell suspensions in a same-day workflow, but also we had fresh frozen material and then formalin fixed tissue for spatial analyses. And so the view was that we could take a multimodal approach by studying single cell genomics, single cell transcriptomics, and spatial biology with immunofluorescence. And so those are the three buckets that you see here in this diagram. And this is really kind of an extraordinary collaboration with the disease management team, who really got on board with this and helped us to collect the tissues and help us to interpret the diagnoses and then ultimately the outcomes of these patients. And so this has led to 3 papers now, and one of which was the one that came out last week, and I'll go over some of that work. So, but I wanted to tell you a little bit about the whole genome sequencing data. So again, this is single cell resolution, and we had been developing technology over the last decade that can single sequence the individual genomes of single cells from cancer, and you can see the readout of this looks something. I don't know if there's a pointer that I can use. Let's see. No. Okay. Well, oh, there we go. Okay. Thank you. Ask and you shall receive. All right. So what this shows is the genome of one single cell. And this is a copy number representation. So on the y-axis is the number of copies of the genome at that particular locus. And then the loci are arrayed on the x-axis, organized by chromosomes. And so what we can see here is that this particular genome has a number of hemizygous deletions shown in blue. It has some amplifications. This is the MYC locus here, so you can see that there's a 8q amplification. So there's a really exquisite copy number readout of an individual cell. Okay, so now we can do this at scale. The platform allows us to sequence around 1,000 cells per cancer. And then when we put all the data into this distribution here, this is representing about 30,000 cells from 40 or so patients. We can see that these cells in this axis, so the x-axis is the fraction of the genome where the major copy number is greater than or equal to 2. And the y-axis is the difference between the major and minor allele. And this is using this allele-specific algorithm here. So we can actually get this information at allele-specific resolution. We can see that the cells really fall into these three groupings. So one is over here, this gray grouping of cells. These actually maintain a baseline diploid architecture. These ones over here, they have undergone a genome doubling, okay? And then these red cells have undergone a 2 times genome doubling. So they've undergone 2 genome doublings. And so we can see that there's very clean split between the baseline diploid and the baseline genome doubled. And what I'm showing you here is actually 2 cells from the same tumor. And so in this tumor, there are coexisting cells with 2 different ploidy states. So you have a diploid representation and a tetraploid representation. You can see that the architecture of the genome is relatively similar. The shape of this amplification can easily be read out from its diploid counterpart, right? So this is the top genome has undergone a whole genome doubling. That's pretty easy to read out. So then if you look at this at the population level, you can see that now every column here is a patient that is in the study. And we can plot that ploidy inference on the y-axis. And you can see that every tumor actually has a distribution, right? And what we found is that every single cancer had at least 2 ploidy states that were coexisting at the time of diagnosis. And so we can no longer really represent a tumor as being tetraploid or diploid. It's a distribution of cells, some of which are tetraploid and some of which are diploid. And so I think that has important implications because in many cases we see considerable populations of cells that are coexisting at both states. And so it's easy to imagine that a therapeutic intervention is going to have a profoundly different impact on the diploid versus tetraploid cells in these tumors. And again, here you have an expanded population of 2 times 2 times genome doubling over here. And so this was kind of a bit of a surprise, actually. And so we wanted to study this a bit. Now, for the purposes of lots of covariation analyses, of course, you kind of have to reduce the patient down to a binary indicator variable. So we called the ones on the left as genome doubled low, the ones on the right as genome doubled high, and started doing correlative analysis on these tumors. Okay. So one of the things that we noted from studying the genomes of the single cells is that in the genome doubled state, the cells that had undergone a genome doubling had introduced a lot of additional chromosomal loss. So what happens is you have a genome doubling event, the cells are unhappy with that. That induces replication stress. It puts has a metabolic constraint on the cells because they now have to carry twice the number of nucleotides. The cells don't actually like it. So we saw is evidence of this gradual loss of chromosomes, and that creates an incredible diversity. And so at the genomic level, we can measure that cell-to-cell variation increased dramatically as a function of genome doubling. So that suggests that there's genome doubling creates evolvability advantage in those cells. So while it puts replication stress on it, the system as a whole has capacity then to be plastic and to evolve in different ways and respond to different stimuli. So we wanted to validate this using multiplex immunofluorescence. And so we worked with Sam Bakoum, who has developed this really nice assay whereby the cGAS protein illuminates ruptured micronuclei in situ. And so micronucleation is a byproduct of chromosome missegregation. And so missegregated chromosomes get sequestered into micronuclei. They have their own membrane. When the membrane ruptures, then that will attract cGAS and stimulate an inflammatory response, the whole cGAS sting axis of cytosolic DNA sensing. So we can measure this, and we basically take these evidence of ruptured micronuclei as a readout of chromosomal instability. And so when we do this in situ for all the patients, we can see that the genome doubled high have a far greater rate of micronucleation than in the genome doubled low tumors. And that's just shown exploded here, where each patient, we can see the distribution of the micronuclei rate. And that's all the genome doubled high ones are on the left. The genome doubled low ones are on the right. And so you can see that there's this really very profound increase in chromosomal instability as a function of genome doubling. So that suggests that there's a real change in how these cells are actually surviving after that genome doubling event. So then we wanted to ask, well, what is this? cGAS is linked to the cGAS sting pathway, which should introduce interferon response and inflammation and drive an immune response. But what we saw is that indeed that's true. When we have this increased chromosomal loss rate, we see increase in sting activity, but only in the genome doubled low. So in the diploid state, this sort of holds true, what you might expect. But once the cells become genome doubled, that relationship actually goes away. And so there's some sort of regulation of sting. We also included sting in this assay, and we can see that the genome doubled high tumors actually have a reduction in sting expression. And so this is potentially a link to immunosuppressive states that we observe in genome doubled high tumors, okay? And so you can then read this out because, again, it's a multimodal type of assay where we actually generate a single cell RNA-seq, including on the CD45 positive cells. And we can see if you compare the right versus left of the volcano plot, the inflammatory signaling is all in the genome doubled low tumors. And over here, you start to have angiogenesis and other types of immunosuppressive expression programs that are classically immunosuppressive, TGF-beta, et cetera. And so genome doubling, through the sting axis, we think is modulating the immune response. And this is all, again, in disease natural history. There's no therapeutic selective pressure here at all. So this is just how the tumors are evolving. Great. Okay, so that's that story. And again, recently published, and please feel free to reference the paper there. All right. So then moving on to the last story. So in addition to pre-treatment collections, we also followed these patients over time through cell-free DNA collections with the idea that hopefully we could track evolution of drug resistance from diagnosis through to relapse. And so part of the protocol was to collect vials of blood, and where we could extract cell-free DNA and use cell-free DNA to infer clonal evolution throughout the therapeutic time course. So just zooming in on exactly the power of the single-cell whole genome sequencing. So what's shown here on the left is a bit of an expanded phylogenetic tree where the terminal leaves of this tree are individual cells. And we can use phylogenetic principles to look at how they're related to each other ancestrally. And so you can see here that in this particular tumor, there are two major clones that can be inferred from the clades of this tree. And when we merge the whole genome data from all the red cells and all the yellow cells, then we can start to reconstruct what does the genome of that clone actually look like. And within each individual cell, there's not enough sequence coverage to actually get to nucleotide level events. But when you merge the data, it's unbiased enough that we can actually start to resolve mutations, structural variants, et cetera. So what's shown now here is, if you can see, these faint gray arcs represent inferred structural variants that represent rearrangements in the genome. You can see this genome is highly rearranged. And remarkably, these gray links are unique to this particular clone. So here it's clone A, and it has these translocations and rearrangements that are only found in the cells that are in clone A. And clone B has its own as well, but you can see here. So each population of cells has its distinct sets of structural variants. And the idea here is that we could potentially exploit this, because this suggests that they create endogenous DNA barcodes. So they're fingerprints that uniquely mark those particular clones. And so the principle that we tried to exploit is that we could track these barcodes over time in the cell-free DNA to identify which clones are patterning drug resistance. All right, that's the idea. So this shows you the overall workflow. So we have the single cell discovery of the structure variants. And again, you can see that we're bringing together two disparate parts of the genome. Here, chromosome 8, chromosome 19. That sequence, that breakpoint sequence, that doesn't exist in nature. It's highly specific. It doesn't exist in normal cells. And so in comparison to a point mutation, which a lot of companies have developed technologies around point mutations, where a sequencing error can easily lead you astray, this is exquisitely specific to those particular cells that harbor that event. So then we can validate, maybe oftentimes we see copy number alterations that coincide with the rearrangement breakpoints. And then the key thing is that we can synthesize capture probes against that breakpoint sequence. So let's see, where's my, yeah. So here we can synthesize an oligonucleotide against this. We design hybrid capture probes. We can do this for, say, 1,000 events per patient, and then try to fish them out of the cell-free DNA. And so we do this, again, for about 1,000 probes and generate duplex error-corrected sequencing down to 1,000x coverage in the cell-free DNA. So we did this for 18 patients. It says 19, I think. In the end, the paper has 18 patients. I think we dropped one for one reason or another. And so one thing to note right at the beginning is that we just try to see, well, what's the clonal complexity from diagnosis to relapse? And what we found is that there's a dramatic reduction in clonal complexity, so that relapse isn't patterned by many, many clones. It's actually, there's a huge reduction in clonal pruning through surgical removal, through chemotherapeutic selective pressure. There are only a few clones that actually withstand therapy. And again, we're a little bit biased because obviously the clones that we're defining are the ones that we can see at diagnosis. We're not doing a secondary tissue analysis where we can discover new clones. However, but from those observations, at diagnosis, we can see that very few actually survive to recurrence. And so this is kind of what it looks like in its full description. So now reducing that phylogenetic tree to something that we can look at on a page. And here you have a diagnosis. This is a tumor with a germline BRCA1 mutation. And of course, every tumor has a p53 mutation. And there are about 5 major clones in this case. So now we project this over time. The bottom of this timeline shows the treatments that were administered. She's BRCA1 germline, so this patient received PARP inhibitors after frontline chemotherapy. And then relapsed, the gray boxes represent when the patient relapsed, and she goes on to bevacizumab and chemotherapy after that. And unfortunately becomes unresponsive to the therapy and has a recurrence. And this is the CA125 serum-based biomarker that shows a representation of tumor burden. Now this track here shows 2 plots. One is the signal that one can read out of cell-free DNA from the p53 mutation alone. And this is a clonal mutation expected to be in all cells, and so you'd expect that this would have a strong signal. But what you can see is that at this time point here, which is the penultimate time point before relapse, the structural variation signal starts to go up, but the p53 mutation is still not registered. So we think that this is not only specific but very sensitive to predicting relapse. And then now you can, at the very top, this is where the most interesting thing is that we can start to see how the clonal complexity changes over time. And so at the beginning, we can recover all the clones. But then after surgery, there's a crash, as you can imagine. There's no signal, no signal, no signal. The signal starts to come back up. And the only clone that's left is this orange clone. So because we have whole genome sequencing, we can start to now ask, well, what's unique about that clone? This clone happens to have a 1.37 kb deletion that restores the reading frame of the BRCA allele, and so restores presumably homologous recombination function. And so that's the clone that that patterns the relapse, and that's a well understood mechanism, but it shows a really nice positive control proof of principle that we can do this clonal tracking and start to interpret what's driving the relapse in this particular patient. So in this case, it's a clone-specific reversion mutation that restores homologous recombination competency. Okay, so then I have a better news story in this particular patient. This is another patient. Here, the clonal complexity of diagnosis is not that big. It's only 2 major clones. Both have the p53 mutation, but one has a clone-specific ERBB2 mutation, ERBB2 amplification, rather. And this is actually a little bit unusual for high-grade serous ovarian cancer, but it does happen. Here we see the genomes of the 2 clones zeroing in on the ERBB2 locus. It's actually a translocation between chromosome 2 and chromosome 17. And you can see there's a very high-level, characteristic high-level amplification of ERBB2, something you'd see in a breast cancer. Okay, it looks just like that. The other clone doesn't have that amplification at all. So when confronted with frontline chemotherapy, what we see is that there's an eradication of the ERBB2 wild type clone. So that gets completely eradicated. When on the first relapse, what we see then is that what's left over is the ERBB2 amplified clone. All right. So then this patient got quite lucky. We happen to have trastuzumab deruxtecan. I know you're thinking about it. You knew it was coming. Antibody drug conjugate clinical trial. And she got enrolled on the trial, and the response is pretty remarkable. So she goes on the trial. And the only clone that's left is the ERBB2 amplified clone. And this drug has been, really revolutionary. And so she has had a complete response and is still alive and well today. And so this is a kind of an example of you can track the evolution of disease. And in this case, we didn't actually, there's nothing that we did to help inform that this patient should go on the drug. But you can imagine a scenario where you could do this, right, and track evolution over time and actually make a decision based on the state of disease as you're seeing it today, rather than what was there at diagnosis. And so I think this is a kind of a powerful technology where, again, it's multimodal. It's using the diagnostic tissue samples with whole genome sequencing, and then using cell-free DNA to make these measurements. You put these 2 things together, you get a very powerful clonal tracking system where in patients, we can start to make inferences about evolution. We've done a lot of this work in PDXs, but to do this in patients is really quite really, I think, very informative. Okay, so getting to the end here. Shown you a few things about the dynamic and ongoing mutational processes of genome doubling. And this catalyzes really therapeutically relevant phenotypic states in disease natural history. And we have to think about how genome doubling is going to impact, for example, the immune responsiveness of the host, and how that's going to drive evolution and evolvability. What I haven't shown you is that in the paper, the drug resistance phenotypes, so this ERBB2 clone and also the BRCA1 reversion, but also many other examples in the paper, they appear to be pre-adapted in natural history. So these are clones that we could find at the diagnosis that had already evolved some sort of mechanism that could explain relapse to in the context of a drug. So this is, I think, consistent with the idea that you have massive diversification throughout disease evolution, and there's therapeutic selective pressure that is selecting out pre-existing clones. To the extent that there's phenotypic plasticity as an adaptation to therapy, we're still working on that, but I think at least we're consistent. This is consistent with the idea that the clones already pre-exist, and so if we could find them and detect them, we might be able to implement combination therapies or bring combinations earlier. I think I'll skip over this, but I think the principle here is that there is evidence here to support evolutionarily aware adaptive trials. And when are we sampling the tumor to make therapeutic decisions? Can we actually read out the state of the tumor prior to making a maintenance therapy decision, for example? In the PARP inhibitor era, that shifted progression-free survival so that patients actually do much better, but ultimately 75 to 80% will still relapse. And that comes in the maintenance setting. So what is frontline chemotherapy doing, for example, to prime those tumor cells for resistance? That's a big unknown question, but clearly that's going to be doing something. And our data suggests that frontline chemotherapy does do a lot of clonal pruning and therapeutic adaptation. So I want to get to something a little bit more philosophical now. So I think that tumor evolution and phenotypic state switching are bound to be constrained processes. So there aren't infinite conformations that a tumor cell can take in order to survive or withstand chemotherapy. And so is that a knowable space? Is it a countable space? I believe that we can try to get there. And if we could get there, can we use large-scale AI systems to help us understand what that space is or how that space might be perturbed by a particular drug? So I think we can ask the question of what data is actually needed to fully learn these constraints and generate predictive and forecasting models that predict future states. And so likely what's needed is multimodal, large-scale single-cell data sets. And they are emerging across the clinical timeline. There are lots of different studies in many institutions. As a community, can we collate and integrate this data to learn the large language models of tumor biology? I think this is something that is achievable if we can embrace team community science in studying these patients at a high resolution. And I contend that we must do this in patients. There's a lot of data in mouse model systems, and that's great, and we can understand mechanistically what's going on. But to understand the relevance in the clinic, this has to be done in patient materials, and it's tractable. It's tractable. Okay. So practice what you preach. All of our data is available. You can use it and study it. All right. So then the last few minutes, then I think everything I've shown you is data limited. And so if we're really going to exploit AI tools, we have to sort of overcome these limitations of that engenders. And so any kind of end-to-end training on one institution's data or on a small-scale study will lead to bias inevitably and lack of generalizability. So, the model will do well on your test set from your particular cohort of interest, but likely won't generalize well to somewhere outside. Then that lack of skill will also limit studying rare cancers. And so we see these rare events, these 1% of cases that have ERBB2 amplifications in high-grade serous ovarian cancer. There's no way that one institution can get enough of those to study that question properly. And so these are some of the limitations, and every place needs to get their data together in order to make it compute ready. And I know there's a lot of efforts here, but I can't emphasize enough how important the data engineering effort is to get this data ready for completeness. And then some of the other limitations are access to compute for large models. So how are we addressing some of these? So I think I've shown you the study on the left, but we have other studies in different disease domains, EGFR mutant lung cancer, immune evasion, and gastric cancer. I could probably list another dozen of these from colleagues at MSK. So we had the idea of maybe we could bring this data together. So every study has, on the order of 100,000 to 1 million cells that we're sequencing with single cell RNA-seq. What happens if we brought all the data together? Can we start to fill in the gaps that are there when we're trying to learn the constraints of tumor biology? So our attempt at this is to try to pull in public data and our own data. And here's just the UMAP of 4 million cells from human tumors. These are all patient studies. And we can start to see some interesting things. And I'm not going to go into the details. This is really more conceptual. But we can start to then ask questions more generally. What pathways are activated after therapy in tumor cells? What are the immunological states in tumors as a function of disease progression? What are the constraints that govern these tumor cell fitness properties? And with enough data, we can start to think about actually mapping this landscape and asking these questions. And so using AI tools, I think, is going to be a really fundamental thing. So what I've done over the last 8 years is build up this program, and we now have 10 principal investigators that are asking various different questions, all computational dry labs at MSK. And so the point is, I think to answer these questions is going to take a community of scientists. These are just some of the amazing work that all these people have done. It's a privilege to work with these people, and I think, we've sort of built up a critical mass of intellectual energy in this space. And so I just show this because I think, having a community of scientists like this creates a network effect that where there really is synergy that goes on, and you get a community that is greater than the sum of its parts. And I've borne witness to this over the last 8 years, and incredibly excited about what these people are doing. And so they of course hire people as well, and so we end up with this really nice community now of 150 people that are working together to solve some of these big problems. This is in New Jersey. You can look across the river and see the beautiful Manhattan skyline. That is where the Yankees are, but that's okay. We won't care about that. But yeah, so this has really been amazing. And so I think I want to say that in order to do this properly, it needs a community. And I think what you have here is you have also different diseases that can be brought together, disease areas that can be brought together to solve some of these problems in human biology in general. So I think that's very exciting. So then lastly, I'll just go back to this picture. So how do we actually complete this? So one effort that I've been involved in, and really it's been exciting, is the Cancer AI Alliance. And so a number of people, colleagues across the US, recognize the same problem that I'm talking about. How do we bring data sets together? And so we got together in 4 major cancer centers, the Fred Hutch, Dana-Farber, MSK, and Johns Hopkins, to form the Cancer AI Alliance. And this has really been an amazing partnership between technology companies and these 4 cancer centers to try to build a comprehensive platform to develop and deploy AI models to accelerate research and improve cancer care. And the idea is that we're using a federated learning approach where data doesn't have to leave the host institution. You can compute models on your local environment and share those models in an orchestration layer. They get integrated and then pushed back out to the local nodes. So that way you can take advantage of diversity in data sets and volume in data sets to start learning these models on much more data than anyone cancer center could do alone. This is an amazing partnership between these 4 cancer centers, as I said, and the technology partners in particular have been a key ingredient into this. So we can't do this without large-scale compute, and they've been great providers of that. So I think it's just one solution, but it's something that could be modeled out to look maybe across Ontario, perhaps across the country, as a way of sharing data in a way that data doesn't have to leave, but models can be trained. So with that, there's a lot of people to thank that touch this work. I won't list them all, but I love working with all these people. It's been a real privilege. My lab is there on the right, and those are particular people that did the work that I showed you today. So thanks for your attention, and I'll stop there.